Hey everyone, welcome back to our continuing discussion of serotonin. This time we're talking about serotonin function, part 1. This was talking about the serotonin functions was too big to fit into one little mini lecture, so I've split it into 2 parts. So let's get started. So first off, let's talk about serotonin and aggression. Brain areas that are involved in aggression or aggressive responses are heavily innervated by serotonin, meaning they receive a lot of projections from our RAF-A system. So for example, regions like the orbitofrontal cortex, temporal cortex, cingulate, striatum, amygdala, hypothalamus, and hippocampus are all heavily involved in aggressive responses, and they receive lots of serotonergic inputs. This is not restricted to just one serotonin receptor type, as these regions express many serotonin receptor subtypes. This has led to the serotonin deficiency hypothesis, the idea that low central nervous system serotonin is associated with hyperaggressiveness. So the less serotonin that you have, the more aggression that we tend to see. Some disorders that are marked by aggression and violence can be helped with serotonin or drugs that are anti-aggression. So things like SSRIs that will increase the level of serotonin that's present will help with aggressive disorders. So here we have some data from a study that your book discusses, in which subjects with problems with violent disorders are given either a placebo or fluoxetine, which is Prozac. It's a selective serotonin reuptake inhibitor, which we've talked about already, will increase the level of serotonin that's present. You can see here we have our baseline period and their mean OASM aggression score, so just sort of survey-based measure of aggressive behavior. You can see here when the treatment onset happens, starting 2 weeks later, we see a dramatic decrease in aggressive behavior, which then rapidly separates out with the fluoxetine group being dramatically lower than the placebo group on their aggression score. And this persists throughout the endpoint of the study. Again, this provides some support for the serotonin deficiency hypothesis, as the aggressive disorder is improved by the increasing of levels of available serotonin. So with all of the regions that serotonin producing neurons project to, it's probably no surprise that it is involved in sleep. We see changes in serotonergic tone during the various states of wakefulness and sleep. Serotonin-producing neurons actually slow down and become irregular during slow-wave sleep. So you can see here during active waking, we have a high level of firing. So each sort of hash mark that is perpendicular to this line represents the occurrence of action potential or increases of firing over time. So this is moving across time, and each little hash mark that's perpendicular here represents activity. So you can see our serotonin-producing neurons are quite active during wakefulness, but become very sparse in their activity and irregular during slow-wave sleep, and are practically entirely inactive during REM sleep. So we see phasic burst-like firing during wakefulness. The thought is that this may facilitate motor output and help to suppress sensory processing, and this also may play a role in responding to reward and punishment. So sort of the classical simple way of thinking about reinforcement and punishment is that's got to be largely involving the dopamine system, but more recent thinking says that it's not just dopamine. There's a wide range of activity that comes into processing reward and punishment, and serotonin seems to be important for that. So we mentioned in a previous mini lecture the practice of generating TPH2 knockouts. And we know a lot about what we know about serotonin function from such studies. So your book does a nice job of summarizing a lot of these patterns. So let's go over those briefly. First off, knockout. This is a knockout of an enzyme that's needed for serotonin synthesis in the central nervous system. Though this does preserve peripheral generation of serotonin. This is important because complete serotonin knockouts are not viable, right? They will not live to adulthood. But preserving peripheral serotonin signaling is enough to keep these animals viable. But they are quite different from their wild type counterpart. For one, TPH2 knockouts are far more aggressive. This shouldn't be surprising given the fact that we talked about how lower serotonin is a predictor of higher aggression. So these knockouts with almost no serotonin are going to be quite aggressive. Likewise, they show maladaptive levels of impulsivity and compulsivity, so their sort of higher order cognition is a little, so they'll make sort of bad or risky decisions and will persist in behaviors in sort of like a habit-like way, more strongly so than their wild type counterparts. Somewhat paradoxically, at least some studies show that they demonstrate less anxiety. They also show poor social communication and social behaviors and deficient maternal behavior. So they don't show the normal social behaviors that mice do, and they are not as good of moms. They are often negligent of their young. They also show impaired thermoregulation, so they're less good at regulating their temperature and response to challenges. Also present are respiratory problems, including apnea. This is because they don't respond to hypercapnia or elevated blood CO2 levels. So they don't have that same sort of chemosensor response that ordinary mice would, because there's no 2A receptor activation, which can cause them not to wake up when they should. This might be a possible cause of sudden infant death syndrome, or SIDS, and sudden unexpected death in epilepsy. All right, next up, let's talk about the role of serotonin in hunger and eating. Are you getting the idea yet that serotonin is a really, really diverse set of functions? Right? We've gone from aggression to sleep to hunger and eating, and we're not even halfway done yet. So with hunger and eating, we're talking mostly about the 1B and 2C receptors, as well as the receptor subtype. So agonists of 1B and 2C and antagonists of 6 will produce hypophagia, or less eating. So hypophagia is the suppression of eating behavior, right? And you can sort of tell that from the word. Hypo meaning less, and phagia, eating, less eating. So one drug that was initially sold for this purpose is fenfluramine. This is a drug that promotes the release of serotonin, and it was used to treat obesity. It did so, but it was pretty non-specific, so it had lots of problems. So it caused heart valve abnormalities and pulmonary hypertension. There were some side effects caused by the stimulation of 2B receptors on the heart valve tissue. Anorectic effects, or the eating suppression effects, were caused by stimulation of 2C receptors, right, as we mentioned earlier, that are found in the hypothalamus. We also have leucocerin, which is used to treat obesity, but is more selective to the 2C receptors, so it doesn't, cause these problems with your heart that were an issue with fenfluramine. It's likely not a surprise to you that serotonin is involved with anxiety. In fact, activation of 1A receptor subtype is the most common target for anxiety-reducing drugs. SSRIs are a common treatment. They, of course, are not specific to a receptor subtype, right? They are specific to promoting serotonin release, but they don't target just one receptor subtype. The drug buspirone, trade name Buspar, is a 5-HT1A agonist, and it is sometimes prescribed for the treatment of anxiety. Mice that are deficient in the expression of this receptor subtype show increased anxiety. You can see over here a figure taken from your textbook, an experiment in which the mice either have a knockout for the receptor, so they lack the 1A receptor, or wild type, which express it. So this particular task is an elevated zero maze. So an elevated zero maze is basically a maze that's kind of shaped like a big donut. So it's got a hollow space in the middle, and this is a sort of a platform that the rats can walk on. And in one portion of the maze, there'll be high walls. So on both sides right there, so there'll be this little thick, high-walled portion. And then these areas right here would be the open areas where there are no walls. And what we measure with an elevated zero maze is how much time do the rodents spend on the open portions versus the closed portions. So mice feel safe in an enclosed space where there's walls to sort of protect them, and they tend to stay in those places when they are anxious. Though with enough time, a mouse will explore the open areas and spend some time there as well. So you can see here that our normal intact mouse is spending some time in the open areas of the maze, whereas the knockouts are spending significantly less time in the open areas, suggesting that they are more anxious. And we also know that agonism of the 2A and 2C receptor types will increase levels of anxiety, and the deletion of these receptors via a similar method will have anxiolytic effects, meaning it has anxiety-reducing effects. Okay, that's it for part 1 of serotonin function. Next time we'll move on to part 2.